my evolutionary topic, I chose to talk about CRISPR for evolution. Okay, so the reason why I chose this topic is because I've been introduced to this gene modifying tool since the beginning of 2019, and it just fascinated me with all the different things you can do with this gene modifying tool exactly. So I recently have decided to become an egg donor, and I feel I'm contributing to science and evolution in that sense to women that aren't capable of having babies, which is similar in a way to this process because with CRISPR, it is possible to even genetically design offspring by choosing specific genotypic and phenotypic traits. Okay, so how this works is CRISPR is also known as designer babies tool, which stands for clustered regularly interspaced short palindromic repeats and is a technique used to edit a cell's DNA. So first, it is a two-part molecular tool, so it uses Cas9 and guide RNA. Then Cas9 is a protein that can make cuts in DNA. So guide RNA contains a sequence that matches the target DNA, which is in focus. The guide RNA directs the Cas9 protein to the target DNA. And so the Cas9 protein unzips the target DNA, which then guides the RNA binds to the matching sequence, so that way they can therefore attach to the site. And this Cas9 makes a cut in the DNA, and once it is binded, at that point scientists can either edit or disable a gene or insert a new DNA sequence. So basically a summary of CRISPR is this. When a virus comes into a cell, they explode and release naked genes. If you're a bacteria, these things can take over your cells, so you must act fast. If the virus is unknown, the bacteria will send out some enzymes, known as ground troops, which fight hard but usually lose and will never be heard from again. More often than not, the virus takes over and the bacteria dies. There is non-zero probability that you survive the attack, and so the resulting bacteria will send out some more enzymes to basically clean up the stray viruses. So this is where storage comes into effect, that those enzymes will take those little bits of virus and store it into the bacteria's DNA right in the little pieces between the repeats, so that way when the virus shows up again, the bacteria will be better prepared to fight off the virus. This is where CRISPR comes into play, because instead of sending the ground troop enzymes and have them die off again, you'll send out the enzymes that know how to fight off this virus. So once this virus is discovered and encoded in the DNA, CRISPR can cut the DNA sequence. So these are the two ladies that discovered this CRISPR in the first place. But first reference to CRISPR was in 1987 when some Japanese scientists were looking at E. coli and found there were 5 repeating sequences, all separated by very short sequences in between them, like little blurbs, which is unusual in bacterial genomes. We now know this today to be CRISPR. Famous quote by Jad Abramrad, sorry if I didn't say that name right. It was directly on a CRISPR podcast that I listened to, and he said that the basic thought was, why don't we turn this defense into offense? Because these things, they seem to be really good at cutting, and yet they only seem to cut the things that are on their mugshot. Maybe I could just replace what's on their mugshot. Instead of them going after viruses, maybe they could go after a gene that causes Huntington's disease or hemophilia. So as stated, this CRISPR usually goes after viruses, but scientists figured, oh, well, we can genetically modify anything we want. So basically the possibilities are endless, and they can modify anything you want to, as opposed to like the mugshot potential just straight for the virus. You can change it to anything. You can change colored eyes, snip that DNA sequence, put in different eye colors, you know? And so as of today, October 7th, 2020, 2 scientists, Emmanuel Charpentier on the left and Jennifer Doudna on the right, have won the Nobel Prize in Chemistry for turning a bacterial defense mechanism into a gene editor known as CRISPR. Although the molecular scissors were discovered 8 years ago, these ladies have finally been recognized for their work. So the results / findings, basically the advantages and limitations of CRISPR is this. For advantages, it is arguably simple and efficient. Considering it can be applied directly in the embryo, CRISPR reduces the time required to modify target genes compared to gene targeting technologies based on the use of embryonic stem cells. Improved in bioinformatics tools and optimization of the experimental conditions resulted in successful procedures that introduced the desired mutation. The limitations include targeted alleles often carry additional modifications such as deletions, duplications, etc., which can be tampered with even when CRISPR is introduced because the scope of the DNA repair system is not to integrate DNA fragments into the genome. Also, secondary unwanted mutational events after target locus could occur. So even though you specifically choose what you want to change, that doesn't necessarily stop the other DNA from acting its normal course. Okay, so in conclusion, in 2012, this mechanism was discovered that DNA interference involving a dual RNA structure that directs a Cas9 endonuclease to introduce site-specific double-stranded breaks in target DNA, which today is confirmed as the CRISPR process. What that basically means is CRISPR/Cas9 genome engineering technology has provided researchers with an invaluable tool to accelerate the generation of embryonic models for biomedical in vivo research. The furious pace of CRISPR development, combined to its versatility and ease of use, have already left a mark in the field of molecular genetics. Its combination with established technologies will greatly expand opportunities for the generation of new and valuable genetically engineered embryonic models for basic and translational research. And these are the references I used. As you can see, the Radio Lab podcast is included there specifically. And I hope you enjoyed learning about CRISPR. Again, my name is Kira de la Cruz, and thank you for watching.